Hello friends, welcome back to my channel Score Points. In the previous video, we have learned the basics of monohybrid cross. Today we can learn how to do a monohybrid cross. So first of all, you have to write the phenotype or the external morphological character, tall and dwarf. Tall is the dominant trait and dwarf is the recessive trait. Next, we're going to write the genotype of the parent. Genotype, it is genetic composition, capital T represents the dominant trait and small t represents the recessive trait. Next, we are going to segregate the gametes. So the diploid condition, from the diploid we have to take a haploid out. We have taken capital T and small t. These are the haploids segregated from the diploid parents. Next, we have to write the F1 generation, the first fenial generations. This is formed by the fusion of the haploid gametes. Now we got a diploid, that is heterozygous dominant condition. Since there is a presence of capital T, it is tall. Next, we have to do selfing of first fenial generation or self-pollination of the first generation, that is capital T small t into capital T small t. Then from this, we are going to segregate the gametes. Gametes, it can be segregated in the same way. We have to take a haploid out of the diploid. In homozygous condition, we have identical alleles, so we have to mention only capital T. But in heterozygous condition, you have to mention both the alleles because it is non-identical. Now we have got 4 gametes after selfing or self-pollination of F1 generation. Now we are going to draw a checkerboard. So for that, I'm writing the gametes. These gametes are segregated by selfing of the F1 generation. For writing the F2 generation or the second filial generation, we have to draw a checkerboard. You should draw 4 columns for the monohybrid cross. Now we have to write the gametes above. That is the gametes which we have segregated from the F1 generation. Now we are going to fuse these haploid gametes to get a diploid condition. Capital T and capital T will get a homozygous dominant condition. Capital T and small t will get a heterozygous dominant condition. Again, capital T and small t, heterozygous dominant. And then we have the small t. So we get a recessive condition. So here you can see it is homozygous dominant, so it is tall character. And next one, it's heterozygous dominant. Because of the presence of a capital T, we can say that it is dominant. The last one is homozygous identical recessive character, so it is dwarf. Using the checkerboard, we have completed the F2 generation. From this, we can write the monohybrid phenotypic and genotypic ratio. So first one, it's the phenotype or the external morphological character. Phenotypic ratio is 3 tall is to 1 dwarf. You can observe that there are 3 tall and 1 is the dwarf character. So the ratio becomes 3 tall is to 1 dwarf. Next one, it's the genotypic ratio, the genetic composition. So you can see we have only 1 homozygous dominant. We have 2 heterozygous dominant and 1 heterozygous recessive condition, that is small t. Hence, the genotypic ratio is 1 homozygous dominant, that is capital T is to 2 heterozygous dominant, capital T is to 1 homozygous recessive, that is small t. So as a whole, now we can see both these ratios. Monohybrid phenotypic ratio is 3 tall is to 1 dwarf and monohybrid genotypic ratio is 1 is to 2 is to 1. Thank you for watching my video. Please like, share and subscribe.